Hello everyone, and welcome to today's Gen webinar entitled Rewiring the Brain, Applying Spatial Omics to Neuroplasticity and Neurodegenerative Diseases. Our webinar today was made possible through sponsorship from VizGen. I'll be your host for today's webinar. I'm Juliana Lemure, Deputy Editor-in-Chief at Genetic Engineering and Biotechnology News, which has been the leading source of information on new tools and technology within the life sciences industry for over 40 years. In today's webinar, our speaker will introduce several single-cell and spatial multi-omics technologies and describe how they can be used to improve our mechanistic understandings of brain circuit plasticity and neurodegenerative diseases. Our presenter for today's event is going to talk a lot more about this topic in just a few moments. But first, let's meet our speaker. Today we will hear from Dr. Xiangmin Xu, a professor in the Department of Anatomy and Neurobiology at the University of California Irvine School of Medicine. We will not only hear about how spatial omics tools are allowing scientists to delve deeper into neuroscience, we will also learn about the University of California Irvine Center for Neural Circuit Mapping, which has emerged as a hub for new technology and resource development in the field of neuroscience, and their efforts to develop single-cell spatial multi-omics tools. Now, before we get the webinar underway, I want to remind our audience that we would love to hear from you in our Q&A session that will begin right after the presentation ends. Xiangmin will be available to answer as many of your questions as he can, so simply type your questions into the Q&A box and hit submit. Okay, with all that said, I will turn things over now to Xiangmin, who will get our webinar started.
Good morning. Thank you for inviting me to giving this seminar. My name is Xiangmin Xu. I'm a professor of anatomy neurobiology and director of the Center for Neural Circuits Mapping at the UCI School of Medicine. So today, I would like to take this opportunity to tell you about our Center of Neural Circuits Mapping and some aspects of our research program. In particular, I would like to talk about our recent focus on spatial omics and their applications in our research of neural circuits and brain disease. I will start to say a few words about my research programs. So we're interested in neural circuits. We're interested in neurobiology of the sensory perception, learning memory, neuroplasticity, and recently we also expanded into aging and Alzheimer's disease research. So we have a broad range of funded projects, and we take a multidisciplinary approaches. We're strong in developing new viral genetic tools for neuroscience studies. And we have been increasing, going big in vivo neuroensembling population imaging, including the in vivo miniscope and the two-photon calcium imaging. We have used spatial multi-omics for our neuroscience studies. And our analysis is refined, informed by further by engineering and computational technology. And our research has significant translation potential. And our cortical plasticity work will help us to better understand the neurodevelopmental disorders, such as childhood visual development disorders, such as amblyopia. And our hippocampus and learning memory rate research, we hope will help us to better understand and treat brain disease, such as Alzheimer's disease. So my lab efforts has led to the establishment of UCI Center for Neural Circuits Mapping. So our center focuses on neural circuit studies and new viral genetic technology development. So we're interested in defining molecular and neurosurface mechanisms that underlie neurodevelopmental, neuropsychiatry, and neurodegenerative disorders. Given the increasing interdisciplinary nature of neurosurface analysis, and our center emphasizes collaborative inclusions of engineering, computer science, and physical sciences faculties on campus and beyond UCI. Yeah. So this is a nice picture. This, so center is relatively young, and this picture was taken at the, when our center was approved in early 2020. And you see, and so these are some of the important people who help to establish our center. And you can tell from here, these are the core leadership of our center. So in addition to our colleagues from school medicine, we're also including our colleagues from engineering school, from a computer science school, as well as from the School of Physical Sciences. So our center members work collaboratively to establish transformative and translational research program focused on our ultimate goal to develop cures for treating human disease of the nervous system. Yeah. And the centers, the membership has grown quite fast. So now we have about 100 faculty members. We have both internal and external members. Internal members have been expanded across multiple schools at the UCI. Yeah. And our external members are important because we're connected through our grants and applications. And so both internal and external members form our broad collaborative infrastructures for our center research programs. So our center research area focus including neurocircuits in health disease. And also it includes the area focus including the viral genetic tool development, gene therapy, and translation research of rare genetic disease. And we also develop single cell technologies for spatial multi-omics and therapeutic discovery. I would like to say that our center has been a focal point for bringing together talented basic and clinical research from different areas toward our common goals. And this actually is important because we form we have strong research teams, right? And so as shown up in these slides, we have assembled strong teams of both basic science and clinical science researchers to secure large-scale fundings for our project. So these slides show our initial success, and that was in 2021. It's very exciting, you say, that we actually received 3 notice awards, and we got award 3 federal grants totaling $7.1 million, including one for $4.8 million grant from National Institute of Aging, and 3 years, a $1.8 million grant from Food and Drug Administration, FDA, and about $500,000 grant from the National Institute of Mental Health to support the efforts of our center investigate to develop powerful molecular tools and enhance resources offered by the center to neurosciences researchers. And here you can see actually a few of the team members are featured here, right, including Dr. Carl Cotterman, and he was a professor of neurology, and our team also including Dr. Tom Lin, he was professor of neurobiology and behavior, and the director of UCI Center for Virus Researches. And Dr. Todd Holmes is my long-term collaborator at the UCI, and our team also including physician scientists such as Dr. Al Laspada and Virginia Kimonis. And Dr. Lisa Head is a professor of pathology, and she's a major leadership in UCI's ADRC. The center also are successful, I would say center members are also successful in large grant application through a broad network of collaborators with other institutions. As shown up here, we will award 5 years, 14 million NIH grant to study brain circuits susceptible to aging and Alzheimer's disease. So we form a multi-institutional, multi-investigative teams, including investigators from UCI, UT Southwestern, UCSD, and Allen Institute for Brain Sciences. So we leverage the strong resources, our center, UCI Center for Neurocircuits Mapping, and the UCI's Model AD Consortium, and as well as the Allen Institute of Brain Science. And so we leverage the investigators' the expertise in neurocircuits mapping, AD mouse model development, as well as the mouse brain atlas development. And so those, and we propose to perform large-scale cell type specific mapping of hippocampal formation circuits to generate serial resolution connectome data that combines molecular and anatomic annotations using multiple AD model mice. Here's another example. In our center now, Sun Team is a key member on a multi-institutional, huge NIH BICAN brain mapping project. So this type of grant success is very good for our center and also for UCI. And this project is to create human brain cell atlas to study individual human brain cells, molecular features, location, and how they change over time. I know a few UCI faculty members are featured here, including Dr. Adam Nugis and Dr. Liz Head, Bill Young, and Craig Starks, Hannah Park, and Rob Edwards here. And now, again, I would like to emphasize the center members are from different areas and really truly work together and through effective partnerships. We continue to expand our center collaborations. We recently award $10 million NIH grant. And the center team is leading multi-institutional studies of single-cell vulnerabilities to Alzheimer's disease. So the multi-institution teams, including UCI, and also including the investigators from University of Minnesota, such as Dr. Michael Ku and the team Ebner, as well as Dr. Bing Ling's team at the UCSD. And also, I would like to bring you to attention to these team members at the UCI. So we're actually including computer sciences faculty, such as Dr. Gobi M, and as well as biological science faculty, Dr. Kim Green, professor of neurobiology and behavior, and Dr. Grant McGregor, professor of developmental cell biology. Very recently, and our center team is awarded $3.8 million from NIH to advance brain initiative research. So this grant actually is a very important grant because this grant has a lot of funding for building infrastructure for our viral production facility, and it will help our center to become a national center of neuroscience resources. And this grant also shows a feature of strong diversity of institutional partnership. So we team up with Dr. Gordon Fisher's teams at Harvard University and the Broad Institute. So we collaborate with them. They put, the Gordon Fisher's teams produce those enhancer AAV regions. So they will transfer the seed region to us. We will, we'll scale up, manufacture, and after quality control, will distribute to the neuroscience community for uses by other investigators. Right, and also collaborate with other less research intensive institutions such as Morehouse School of Medicine in Atlanta, Georgia, and the Western University of Health Science in Pomona, California. And so this type, I think this kind of grants is really important to allow us to again build the broad infrastructures. Right, and also really, so to fulfill NIH the missions or expand inclusiveness in neuroscience community. So by leveraging the federal funding and our local support, particularly UCI school medicine support, so we have established our strong infrastructure and critical resources. And so if you go to our website, if you click resource and service, you can actually see we have actually offer very strong resources and service to our UCI investigators as well as external investigators. And I would like to really like to showcase a couple of the facility services. And the first, so this is also say that this slide also show that is the UCI the centers has quickly emerged as hub for new technology development and resource generation at the UCI. And shown up here, our viral core facilities support basic and clinical investigators using viral genetic tools and serve UCI research communities by custom making genetically modified AAVs, NDs, and neurotropic viruses such as rabies and herpesviruses. So in addition to neuroscience reagents generation, part of the project is evolving to develop new therapeutics. So our recent work is focused on single-cell genomics, epigenomic analysis of brain circuits in health and disease. And we're using the latest single-cell technology coupled with functional circuit mapping and behavior analysis. Our goal is to determine how epigenome shapes neurocircuit activity and the learning memory behavior that change with aging and AD progression. So using the latest single-cell technology coupled with functional circuit mapping and behavior analysis, our goal is to determine how the epigenome shape neurocircuit activity, how epigenome influence, how genomics and epigenomics influence learning and memory behavior that change with aging and Alzheimer's disease progression. And these research directions really benefit from ongoing multiple funding collaborations with Dr. Bingling's team at UCSD and model AD investigators at UCI. So we present the conceptual advancement and new approaches to study the brain disease in our recent published expert review articles in molecular psychiatry. If you're interested, you are welcome to go there. You're welcome to check our paper. We have made good progress in this direction. And our single-cell epigenome analysis reveals aging-associated decay of heterochromatin domains, particularly in the excited neurons in mouse frontal cortex. And recently, we also our team also discovered a novel inhibitor cortical neuron type, and that this cell type is implicated in neuropsychiatry conditions. This paper is also recently published in Molecular Psychiatry, and so it's very important for us to use the multi-omics, and particularly use 10x multi-ohm to examine both single-cell RNA and ataxis profiles of these specific cell type and other cell types. And this will provide us the molecular mechanisms of the potential function of these cell types in the brain. Our center also start to build strong single cell and spatial multi-omic resources. As shown up here, we have acquired 2 visiting microscope instruments. And we have been successful running first experiment on human, mouse, and tissue. And so not just, so we, as shown here, we actually run the merfish, this is merfish data from the human temporal lobe coronal section. And so this is actually beautiful data. This is from the brain section. So you can see those color coded at different cell types, right? And so I orient you, these are the surface, right? The surface. And as you can see, this is the superficial cortical layers, layer 2, 3, right? And then it goes to deeper layers, showing different color blue. And this is white matter area. So this is showing down here. We have been successful on the brain samples and also the tissues from peripheral systems. And so the most issues really has been powerful approaches in our recent studies. So just like in summary, what I tell you so far. Told you so far. So with our strong collaborative infrastructure, so we have successfully assembled highly talented collaborative groups that including both basic and physician investigators. And we have secured major fundings at the very highest level to take on big scientific problems. Our center has emerged as a hub for new technology and resources for biomedical research at the UCI, and the center provides multiple services and resources. And so I believe our center infrastructure and resources will help to position our team toward establishing a top national translational neuroscience research program at the UCI. So in the next, I will go to the specific focus of the single cell and the spatial omics. Specifically, I'm going to introduce our recent work of single cell spatial transdomic analysis of Alzheimer's disease pathogenesis in mouse models. So this project is Model AD project. I have listed my co-author's name here. So for this project, we apply merfish. We use 300 gene probes. And we use merfish to measure 300 RNA species in single cells to produce large-scale spatial resolved gene expression atlas generated from 5X80 and age-matched wild-type mice. Yeah, 5X80 transgenic mice has been widely used in the field, and these mice are good because they replicate the new pathological features in human Alzheimer's disease. And in addition, we investigate the effects of TREM2 R47H mutation. This is yes, I will tell you a little bit more in next slides. So this mutation is a strong risk factors for development Alzheimer's disease in humans. So we use this mutation mouse model produce the atlas for the TREM2 mutation mouse along or crossbreed of 5X80 and TREM2 R47H mutation mice. So GWAS study have been used to identify genetic variants associated with Alzheimer's disease. So one key discovery identified in the TREM2 gene was R47H missense mutation, and it has been identified as a strong risk factor for development of late onset Alzheimer's disease. TREM2 stands for triggering receptor expressed on myeloid cells. It acts as a receptor for binding the beta amyloid and ApoE. And this receptor is required, you can tell from this illustration, is required for microglia activation and eliminate cellular depletion. So recently, our colleague at the UCI Model 80 have produced a new mouse model with the TREM2 R47H mutation. So this mouse model is a good model, good mouse model. But this mutation alone is not sufficient to induce amyloid plaque pathology in mice. So we consider cross the 5x80 with TREM2 mouse model. And so then we can examine the TREM2 mutation impact compared with age-matched 5xF80 TREM2 mutation mouse and wild-type controls. So we reason that the mouse exhibit both 5xF80 and TREM2 mutations, by studying them, and it will help us to understand the transcriptomic influence of these mutations in developing Alzheimer's disease-like pathology. So this slide shows our experimental data information. So as I told you, we use 4 different mouse strains: wild-type, 5xF80, TREM2 mutation, and TREM2 mutation cross to the 5xF80. So we have 4 genotypes, and we collected single cells from 19 clonal slices. So each genotype will have 3 to 4 sample slices, and we use up a total 15 animals. So the Murphy experiment is really have a high cell yield, and so we actually based upon these 19 clonal slices, we actually yield over 400,000 cells after quality control. The previous studies have analyzed spatial transcriptomics with neurodegenerations in aging, in microglia activation. So this is good because it's improve the efficiency, effects, the spatial approach such as Murphy in investigating the transcriptomic dysregulations in the brain. However, the previous attempt to analyze spatial molecular impact of Alzheimer's pathology in mouse models has been limited, either in cell type, resolution, or the numbers of cortical regions. So that's why the Murphy is a powerful tool to generate for our Alzheimer's disease research program. So as shown up here, you can see that this is a video I taken from the Vision company website. As you can see that you can zoom in, so the Murphy should really have a very good resolution. So you can real imaging and measure individual RNA transcripts. So basically, tiny little dots shown up within the cells. You can measure individual transcripts in several resolution, subcellular resolutions. And also you can look at it in this case, you show that the whole clonal slices, right? You can see actually look at or multiple, at least a dozen of cortical and subcortical bone regions here. So this will allow us to generate in large-scale spatial resolved molecular atlases. And MOFISH, another thing, the MOFISH is, stand for multiplex error robust fluorescent in situ hybridization. It's a fisher technology, but does has very cool, the error correction, mechanism. Here we present a single cell resolution spatial transdermic atlas of 5xAD and TREM2 mutation-induced alteration across the cortical and subcortical brain region in mouse clonal sections. Yeah. And the examples are taken from one of the 5xAD transgenic mouse samples. So this is the half of the clonal brain slice. You can see this cortex and subcortical region, including hippocampus and thalamus and midbrain, and also including some of the subcortical cortical amygdala regions. And so you zoom in, and so actually see, so in this case, I told you this, we use 300 gene probes. So different colors now indicate different the transcript, RNA transcripts. So these MOFISH samples are analyzed through our custom pipeline, and samples are integrated across batches. As a result, are clustered and reduced 2 dimensions through, as shown here, like UMAP here. And the cell types identified use a combination of canonical cell markers as well as their spatial positions. Right. So because the spatial, it is basically spatial analysis, the whole cortical brain slice, you can have in this case, actually, we can divide it into a dozen of the brain regions. And so generally, the conventional single cell just give you cell type, right? Cell clusters here, right? But now with the spatial analysis through the MOFISH, we'll be able to really get to the specific cell types, right? We actually map those cell types to the spatial biology, right? So you can see in these cell types are color coded, and so the color coders are the exciting neurons, and also the gray scales we actually we map those non-neuronal cells or the inhibitor neurons. So those are those showing the lamina specific features. And so if we look at those the layer 3 cells, they actually yes, they actually indeed they are distributed in the superficial cortical cortex here. Right? And then also you can see the different subfield, the hippocampus. This pink area is a thalamus cell, yeah. And the cortical amygdala are shown up here. So these are very rich data sets, right? Because you have like here you could up to like 30 more than 30 cell type. You have dozens of different regions, right? And if you multiplex them, there's a lot of rich information to be mined out. Again, I show a little bit more of data overview here, and those are beautiful data maps here. And so color code, same color code scheme as shown in the last slides, and we can really look at. So here showing the different columns are different genotypes, right? And so for each samples, we can look at the cell type proportions, and they relate to the spatial distributions as shown in different pie charts here. So now let's look into our data details. And shown here, and MOFISH of the clonal brain slices enable analysis of the spatial distribution of individual genes. So as shown here, this visual, we visualize the transcripts of the TMEM119 and ITJX shown here. Right. So yeah, so those are the TEM1s are shown in green, and the ITEX are shown in red, right? And so we can visualize, we can see the actually there's upregulation of ITGAX in the 5xAD and TREM2 cross to 5xAD mouse samples. In the bottom row, we also visualize TREM2 expressions. So the Y type, TREM2 alone, but in the 5xAD and TREM2 cross with 5xAD, you actually see much enhanced TREM2 expression. MoFISH also allow us to enable spatial analysis of local effects of AD pathology on the regulation of gene expressions in nearby cells, as shown in these slides. So these slides, so those are the DABI slides, right? And on top of that, you shown the plugs. So we actually use the machine learning approach. You can identify those beta amyloid plugs shown here. Those are color-coded ones. Yeah. And so this is a whole brain slice. We use cell pulse. Right, it's one of the recently developed machine learning algorithm. So you can really automatically pick up those plugs based upon its DABI staining. And so those plugs are quite different from those nuclear staining, nuclear DABI staining. Right. And then, so for the so here summarize some of the data compare with plug densities in different brain regions across two genotype 5xAD and TREM2 cross with 5xAD. And in general, I see it's relatively similar, but we do see some regional differences, particularly in neocortical areas and midbrain and Alzheimer's. So the spatial identification plugs enable analysis of plaque microenvironment in terms of their cell type distributions and gene expression. So I will introduce these slides a little bit more here. So first, let's look at the images here. Right. So these we identify. This you can tell we have the beta amyloid plaques here, and those are regions. Those are segmented the cell type, the cells. Right. So this I believe this is the a little portion areas from the CL1 hippocampus CL1 areas. And those are green circles are shown here, microglia. So this actually show a lot of microglia, right? So we already know that this is actually expected. We know that there's a lot of other microglia associated with the plaques. And so that's why when you do the proportion of cell type close to plaque, you see 60% times you find the microglia close to plaque. And next one you can see here is those are astrocytes. Those are pink, circle areas. So those are astrocytes, and then other glial cells. And actually, it's interesting. You actually see much fewer, oftentimes much fewer chance you see the neuronal close to microglia. And so this is just look. So because MoFISH is really spatial. It's really conduct spatial biology analysis, right? So with spatial, so you can look at the AD pathology and look at how the distribution cell type, differential cell type distribution with close to this pathology, and then also we can look at how the gene expressions patterns altered and close to plaque or away from plaque. So these exactly those some of the results, examples results are shown in these slides. And so our examination in terms of the plaque plasticity is showing that there are differential gene expressions. And so we do two analyses. One, we just look at the continuous distance from a plaque, look at the microglia and the astrocytes gene expression analysis, right? Because we can identify the cell type, this specific cell type, and look at their gene expressions. Another approach we look at is just look at differential gene expressions and testing the cells within the center, within kind of close to the center, like 100 within 100 microns, versus of those distances beyond between the 100 500 microns from the center plaques, and so we'll be able to see the differential. Basically, look at how the cell type specific gene expression is impacted by plaque relative to their closeness to the plaque center. So those data show the microglia cells. When you getting closer, of course, you see more of the disease associated microglia gene expression features. And similarly, there's also more astrocyte reactive disease associated astrocyte gene expression profiles. And interestingly, and also this allows us to look at neuronal gene changes. So in this particularly, we found that the layer 6 cortical deeper cortical layers, layer 6B excited neurons, the superficial cortical layers, layer 2, the IT neurons intraterecephalous excited neurons. And also we see some of the inhibitor neurons gene profile change, including the parabolic cells. And then you can see the cortical amygdala excited neuron change. And there's also some changes in the layer 5 non-terminal excited neuron change. So with all the type of data, and we can perform some of the qualitative comparison. In this case, I'm showing here so here this slide shows our data indicate the microglia astrocytes. They show the 5XAD induced transcriptional change, but shows less impact from the TREM2 mutation. As I told you, so we use 4 mouse genotypes for these studies, right? And so we can do 4 pairwise comparison, right? So so those are if you look at the heat maps here, so you have 4 columns, and 1, 2, 3, 4 correspond to 4 different pairwise comparison. So let's look at its the A portion here. This shows the pairwise differential expression between genotype among glia population. So A shows microglia here, and astrocytes shown here, and also have oligodendrites and oligodendrites precursor section. So you can tell that microglia astrocytes show lots of 5XAD induced transcriptional change. So these are 5XAD versus wild type, first column. Second time is TREM2 mutation versus TREM2 mutation caused with 5XAD. So you see a lot. So and here is different rows indicate the significant change in genes. Okay, and you see there's a lot of red. So those color code are showing up log for changes. So there's a lot of significant changes, right? High magnitude changes for the 5XAD weighted comparison. In comparison, well, yeah, the third column is for TREM2 with wild type. Fourth column is TREM2, 5XAD versus 5XAD. So basically, look at TREM2 mutation impact. Well, you see some of the changes, but the impact seems much less compared to the 5XAD dependence. And these generally apply to astrocytes. You see astrocytes, so 5XAD columns and TREM2 columns. Yeah, and it's actually real. So the blank areas are non-significant, right? Non-significant. So we see relative few changes in terms of TREM2 mutation impact. And we can also now perform the subclass analysis as shown B, right? So microglia, we actually based upon our samples, we actually will be able to subclass into 7 different cell microglia subclusters here. And if you run, and here shown the pseudotime analysis here, you can see some transition from this cell type which gradually transfers to different. So this basically shows the non-bifurcate the differential transition here. And in terms of these clusters, we're seeing the 7 clusters, subclusters. So we believe the cluster 1 through 5 are more disease associated, and the cluster 6, 7 is more homeostatic related microglia. And they do have associated, yes, you can tell from here, those are proportional abundances here. You can clearly see that homeostasis is actually more associated, is expected associated with wild type or mutation alone, but 5XAD cause mice show much more of disease associated microglia. Yeah, and here showing some of those clusters have some differential or regional specific features. And so number 5 is a very similar feature, like shown in number 3, the 3 channel. Similarly, we also look at the apply subcluster analysis and perform some of the pseudotype analysis as well. Clearly, and you have some disease associated. This is similar to microglia. You also see disease associated and exercise more close, more to the 5XAD reached mouse samples, much less to the Xiangmin mutation sample. Because the morefish, as I told you, morefish is nice thing that morefish will be able to look at the cell type specific and gene expression changes in spatial contact. Right? And also we can look at where those are disease associated microglia exercises are distributed in the in growing slices. So this is exactly shown. Some example are shown in these slides. So we found that disease associated microglia are enriched in the corpus callosum and some of the subcortical regions. And also we found the disease associated astrocytes are localized almost exclusively to the corpus callosum and its surrounding areas. So let's look at some examples. So this is one of the examples shown that here those disease-associated microglia are shown in blue, and homostatic microglia are shown in yellow. So as you can see, that is a lot of class those disease-associated close to the corpus callosum, these corpus areas, and also in lots actually is quite a bit of in the midbrain itself. And if we look at the disease associated microglia proportion of the overall microglia population, you can see the highest is the corpus callosum CC areas, and also you see high in the midbrain and the thalamus area, right? And so this in terms of 5XAD. So interesting that if you look at the samples from Xiangmin Xu across the 5XAD samples, right, those are shown in green. You see some region specific differences here. And then we can also compare these two genotypes, look at those microglia distributions in different cortical layers, right? Because we know that you can clearly see difference here, and so it looks like that the low deeper cortical layer 5, 6 has more disease associated microglia as shown here. Right? Although within genotype, we don't between genotype we don't see much. But if you do see the lamina specific differences here. So we can apply the similar analysis to look at these associated astrocytes. Right? So disease associated astrocytes are highly concentrated in the corpus callosum in surrounding areas. And so yes, this quantification shows very high in terms of proportion wise. And this disease associated astrocytes are also shown the lamina specific distribution differences. Right? So after we look at microglia specific gene expression or spatial distribution changes. So now let's look at neuronal rate gene expression change. So early studies, in Alzheimer's disease, research using 5XAD or other mouse models been looking at mostly been focused on to look at the microglia activation, and some or do look at astrocyte activation, but neuronal alterations has really not been examined. Right? And so and we actually found that these are actually some new findings we found in our current studies. They are shown here. We found the layer 2-3 IT and including some other exciting neurotypes such as layer 5 and non-pyramidal NP excited neurons exhibit consistent Xiangmin 2 associated transcriptomic variation. And and some of the subclusters are spatial localized in showing the genotype biased. So let's go over this important slide. Right. So again, there's a way, because we're looking at 4 genotypes. So 1, 2, 3, 4. 1, 2, 3, 4 correspond to 4 pairwise genotype comparisons. So the number 1, 2 that mostly look at is 5xAD rate differences. And the number 3, number 4 columns we'll look at the TREM2 impact, TREM2 mutation impact. And so now you can see that it's different from the you've seen before. Now you see more warm colors, more those grades show warm colors when you will compare with TREM2 rated genotype. So I want to point out some interesting gene expression differences, particularly they're called some interesting one, including here you can look at is BDNF, and those BDNF rate receptor, the NTRK2, basic TrkB receptor gene changer, and particularly the TREM2 the NTRKs actually consistency shown into the other cell types, including layer 5, IT, right? And also layer 6, IT. So you look at the warm colors here, and as well as is layer 5, NP, NTRK2, right? So we basically found the differential expressions in cortical exciting neurons, and often identified the NTRK2 as upregulated, induced by TREM2 mutation. And the NTRK2, the TrkB, is known as strong impact the MAPK signaling cascade required for synaptic plasticities, including the LTP. And then we perform other, we identify those interesting cell type, and then we can do some further subclusters. Right? So if you look at L2IT cell types, right? And those cell types, they're interesting because those types, if you look spatial-wise, they only, really like the very superficial, like layer 2. That's why the layer 2 are the mouse, cortex. And they are associated, if you look here, they actually, so this is a proportion change. These are the genotypes, right? And so the light colors indicate they have strong presence in the Y-type TREM2 mutation. And they are much less in the 5xAD rated genotypes. And another example is layer 5 NP excited neurons. And so those are cluster cell types. They actually, so the blue ones, showing the other. So this is generally those layer 5 non-primary are relative sparse. But look at the class 2 cluster here. The class 2 actually showing up is really close to some kind of pre-subiculum or the retrospinal cortex. And the other cell type is actually located in the layer 5, middle layer 5 in neocortex. Right? And it's also highly associated with Y-type TREM2 mutation, but not so much with 5xAD and 5xAD cross with TREM2 mutation. So the subclustering of cortical cell types identify genotype-specific subgrouping with unique marker genes and spatial location. So to sum up for the Murphy studies, and so we believe this is actually quite interesting, also highly significant. And our studies, first we demonstrate to enable a single cell brain-wide analysis of transcriptomic dysregulation. And spatial analysis identify unique glial and neuronal transcriptomic alterations induced independently by 5xAD and TREM2 mutations. Now we believe those transcriptomic alterations can impact the inflammatory responses in microglia and astrocytes, as well as neuroplasticity and the learning memory associated molecular responses in neuronal cells. I will not go to those detailed, these specific details, the insights we gained from these studies. Right? But I would like to say that the recent progress in single-cell, spatial, and spatial omics allows for determining the identity, location, and circuitry of specific cells affected by neurodegenerative disease, including Alzheimer's disease. So the newly developed technology, including MOFISH, and also the conceptual advance that we propose, right? So we present new opportunities for study and treating the circuits vulnerable in AD and other neurodegenerative diseases. Since Alzheimer's disease is just one of the type of the neurodegenerative disease, right? And recently, the center members also have been interested in to apply the MOFISH to study Huntington's disease. Huntington's disease is a neurodegenerative disorder that primarily affects striatum region of the brain. So as shown up here, right? So we run the MOFISH experiment. So as you show here, we have some beautiful data, preliminary data shown here, right? So one thing I just want to notice that it's so nice because the MOFISH is you've seen the MOFISH, the movie, right? So you can look at the very high resolution individual transcript, and then also you will be able to, because it's a relatively large imaging area, you can cover the whole coronal slice in that movie, right? And in here we actually do the MOFISH using the parasagittal slices. Right? In these slices, actually, you can cover from cerebellum through some of the striatums, some of the frontal cortex. You can cover more than 20 bone region in these parasagittal slices. So this is really large scale, high resolution, large scale imaging approaches, and really allow us to accomplish our objective for the cell type specific differential gene expressions. To compare wild type mice shown up here and the Huntington's disease transgenic mouse sample shown here. So we currently we're use our established approach pipeline and to processing these data and to see what's the potential molecular mechanisms underlying the Huntington's disease in these sets of studies. So I would like to thank the Visagen's invitations to give this presentation, and I should say that the Visagen has been we have great collaborations with Visagen's. So we got two systems from we're really the early adopter of the Visagen's most scopes, and it's actually very important to acknowledge their help with our initial setup. In addition to that, Visagen has been has extensive collaborations with our center, including sponsoring some co-hosting the important events, including, so we had a very successful 2023 center conference, which was joint sponsored by Kharkov, Allen Institute for Brain Science, and our center. And the Visagen team came to work with us to present the spatial transdermic workshop, and also they work with us to offer the conference attendees some hands-on training experiences through the Moscow bootcamp. Those are important, very important outreach events for technology dissemination. So those events are well received. I think it definitely will have a good impact for the field. And these I just want to say that at the UCI we have received very strong support, as I mentioned to you earlier on. But this is, I just want to say that we have a very nice new building here, and our single cell spatial omic facility is located in this interdisciplinary science engineering building. And finally, I would like to thank people working in my lab, and I have a wonderful, I have a wonderful, really wonderful team here, and the students, postdocs, and other staffs are highly motivated. And I also would like to acknowledge, I would like to thank our collaborators. I do have a large number of collaborators. Their help, their support, collaborations are essential for our success. And I would like to acknowledge the very generous fundings from NIH, FDA, and other funding agencies. Thank you, and I'll be happy to take questions.
Thank you so much for that, Xiangmin. What a great presentation. I can hardly wait to delve into the questions and continue the conversation on spatial omics. All right, awesome. Well, we're just going to jump into these great questions coming in here. So let me start, Dr. Xu, with this kind of broader question before we get into some of the details. Can you please further comment on the impact of the methods and applications for single cell and spatial multi-omics? And you're just, oh, perfect.
Yeah, thanks, Julian. I think this is great. Yeah, it would be great to make a further comment. So I believe the technology advancement and offer new tools for scientists like us to really address the critical questions. So as illustrated in my talk, I show that the single cell and spatial surface-focused omics really allow us to determine the identity, location, as well as circuitry of specific cell types, which was a factor in the neurodegenerative disease. And these newly developed technology indeed present new opportunities to study and also treat those brain disorders, which I truly believe so. And if you look at the field, there actually is tremendous progress in the last 5 or 10 years in the field of omics. And this new technology, particularly the spatial multi-omics approaches of the joint analysis, not just one modality, it's really multi-modalities, including genome, epigenome, transcriptomic protein, as even potentially including the metalome from single cells. So I do think that these recent technology advancements are starting to transform our understanding of the neurobiology in health and in disease. And so I also, as I foresee that the methods and applications for single cell in spatial multi-omics will enable the new insights into both structural and functional interplays and between intercellular molecular mechanisms that govern the developmental physiology and pathogenesis. So that's my further comments. Thank you.
Okay, terrific. Thanks so much for that. Now, I think we're going to get into a little bit more specific questions for you here. So one of our audience questions says, for segmentation, which markers were used? And they say, microglia have different morphology. So which combinations of markers did you use to get good segmentation for microglia cells, neurons, astrocytes, etc.?
Okay, so that's these very good technical questions and related to the cell segmentation. That's very important. Right? If we could not done the segmentation right, you will have issue with data analysis, data interpretation. And so for the Murfish, for the commercial platform, the Merscope system. So that we have the staining, DAPI staining, and poly A staining, And sometimes you could have them cell membrane staining, a boundary staining, right? And for the brain slices, particularly for those mouse brain slices, I think we segment the cells based upon the DAPI staining and poly A, so basically total RNA, content staining. And there are two ways. Normally, there's two ways for us to do the segmentation. So we start with the researchers, the platform, the software called Merlin. I think they have the commercial version of the Merlin software. And more recently, we've been adopting have adopted the machine learning based approach, cell pulse. Actually, these actually work quite well. And so based upon, so we actually don't use so basically just based on DAPI and poly A staining and use the machine learning approach. It's actually worked very well. You can we did benchmarking and validation, so there's no issue. Really, there's no major issue for us to distinguish different cell types, right? But on the other hand, there's certain things you need to be careful about, because astrocytes or microglia, there are a lot of processes, right? And only segmentation really is focused on the somatic region, so that's something important. And also there's certain, just like a certain single cell sequencing, you may have contamination from other things. So we need to be careful about the background. You could potentially there's astrocytes or microglia processing getting into the neuronal signal. So there's some we have developed a certain technique approach to deal with those issues. Thank you.
Okay, terrific. Thank you so much. Another question from an audience member says, they're a person new to spatial transcriptomics. So they said, thank you so much. It was a great presentation. And they asked, have you observed significant batch effects when running the same set of samples on different slides? If so, could you please share how you've addressed this issue?
Okay, great. So this is another great technical question, batch effect. That's very important. So I should say that actually was to my surprise. We actually found the Murphy experiment use the Moscopes. We have very good robust stabilities across experiment. And actually, I have not seen a major I see that the batch effects, seen from the Murphy experiment seems much less than the conventional single cell sequencing. Right? So there's many reasons. One thing that I believe that the Murphy, you just put this you do the brain slice, right? And there's no involve less, they're not involved with cell dissociation, other things. And so what we did actually validate in we start to use the Moscope system probably about 2 years from now, right? And in first year also we've been done a lot of validation experiments. So what we do that we section the brain slices, where you take two agents, two next to slice next to each other. You run one slice, 2 weeks ago, and another one 2 weeks later. And you just look at visualize those cell clustering or the spatial location. There's basically there's some almost identical. And so this is something I think is about important is anything is not easy to work out. Once you get all the technical details worked out, the batch effect is not so major, and it's just quite stability. That's something I really enjoy. Thank you.
Okay, and I think we have time for one more question, and this one just came in from an audience member's. How do you anticipate therapeutics will be impacted by your findings? It's a great question.
Yeah, that's a great question. So that's exactly what we want to do, right? Because when yes, we want to do basic research on cell mechanism. Eventually, we would like to impact human health care, right? And I should share it. So, the example example research I present here is one of the major neurodegenerative diseases, Alzheimer disease. Right? And so Alzheimer disease. So one of the things in so I generalize first thing that because with the spatial that's very important because conventional single cell sequencing, yes, allow you to see cells, but you just don't know where, what those cell types exist. Right? For example, you just could not to look into the molecular alterations for those cells next to plaque versus far away from plaque. So this actually, I do think this is one of the strong points I would like to make in my presentation. So with an analysis with spatial preserved slides, you'll be able to look at the circuits, those alterations within neural circuits focus. And so far we've only done, single slides, but there's a new technology developing in lab, we should be able to make essentially, high density sampling through the whole brain, perhaps in the whole, those hippocampal formation circuits. And back to us, the therapeutics that so we, in addition to the mouse experiments, so we've also been run parallels, with tissues from human tissues, including the patients. Right? And so I hope that because of some of the things that we were interested in, we found that we have now we allow us to get potential molecular targeting, so including particularly if based upon our the finding when we come up with certain therapeutic, particularly work against neuroinflammation pathways. That's actually, I think, it could potentially translate into the clinical treatment. Thank you.
Okay, awesome. And just in our very last minute, I'm going to have to ask you to do this one, and we're under the gun here, but can you comment on the industrial and academic research collaborations in light of your experience with VizGen?
Oh, okay. So that's something, yeah. So first I have to say that, thank VizGen team. And we actually, it's, as I told you, we're the really early adopter of the VizGen system. And the team from both sides actually work very well, very well. And so it's a really great experience. And in light of our academic research collaboration, so I should say that there should be, these type of interactions, collaborations should be, highly encouraged whenever possible, right? And because I think it is mutually beneficial. In VizGen's case, although we have VizGen system, but we have some unique strengths in academic settings, right? So we, actually the interactions both ways. Sometimes we have issue with their, system, hardware, software. They help us fix. On the other hand, we can provide certain adjustment, improvement in terms of the sample preparation, processing, other things, right? And and I do think generally, it's although there are some, people may ask about how you manage the IP property and other things, but for the scientists, particularly for academic settings, this royalty is not a major constraints for us. I do think the academic and research industry academic collaboration will be able to stimulate and also strengthen the scientific research and technology development in both ways.
Awesome. Okay. And the collaboration going on at your center just sounds totally amazing. And unfortunately, we have to end there. We could keep talking about this all day, I'm sure. But with that, we're going to have to end our webinar. I'd like to remind everyone that this webinar will be archived on the Gen website. That's at genengnews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward it to any of your friends and colleagues who you think might be interested in this content or the webinar itself. I'd like to again thank Dr. Xu again for this incredibly informative presentation. I'd like to thank you, the audience, for your attention and some really great, thoughtful questions. A very special thanks goes to VizGen for sponsoring this webinar, and hopefully we'll see you again at another Gen webinar in the near future. So goodbye for now.